Your hands are a perfect example of something simple yet profound. You have a left hand and a right hand. They are mirror images of each other. They have the same parts, a thumb and four fingers, arranged in the same order. But they are not the same. You can't put a left-handed glove on your right hand. It just won't fit. You can't lay your left hand perfectly on top of your right hand, palm down, and have them match. They are different. This property of being a mirror image that cannot be superimposed is called chirality. It comes from the Greek word for hand. And it's not just for hands, it's everywhere in the universe. Screws are chiral. A standard screw is right-handed. You turn it clockwise to tighten it. A left-handed screw would be its mirror image. You turn it counterclockwise to tighten it. They look almost the same, but they function in opposite ways. The world of molecules is full of this handedness. Many molecules can exist in a left-handed form and a right-handed form. These are called enantiomers. Chemically, they are identical. They have the same atoms connected in the same sequence. They have the same weight, the same melting point, the same boiling point. In a normal lab, they behave in the exact same way. If you synthesize a chiral molecule in a test tube, you almost always get a perfect 50-50 mixture of the left-handed and right-handed versions. This is called a racemic mixture. It's what you would expect from pure chance. The chemistry has no preference for left or right. But biology is not like that. Biology is biased. Life, as we know it, made a choice. When you look at the fundamental building blocks of life, you find something strange. They are all one-handed. Take amino acids. These are the small molecules that link together to form proteins. Proteins do almost everything in your body. They are enzymes that digest your food. They are the hemoglobin that carries oxygen in your blood. They are the collagen that makes up your skin. There are 20 common amino acids that life uses. Each one, except for the simplest one, glycine, is chiral. They can be left-handed or right-handed. Yet in virtually every living thing on Earth, from the smallest bacterium to the largest blue whale, all the amino acids used to build proteins are left-handed. They are L-amino acids, the right-handed versions, the D-amino acids, are almost completely absent from the machinery of life. It's the same story with sugars, but in reverse. Sugars are the building blocks for carbohydrates. They are used for energy. They also form the backbone of DNA and RNA, the molecules that carry our genetic code. Sugars like glucose, ribose, and deoxyribose are all chiral. And life, in its wisdom, chose to use only the right-handed versions. They are D sugars. The left-handed L sugars are basically ignored by biology. You could eat a spoonful of L-glucose and it would taste sweet, but your body wouldn't be able to use it for energy. It would pass right through you. Your cellular machinery, your enzymes are like right-handed gloves. They can't grab onto a left-handed sugar. This is the central mystery. Why? Why this incredible consistency across all of life? Why only two handednesses, left and right? And why did life pick one of each and stick with it so rigidly? It's not that L amino acids are somehow better than D amino acids. They're not. A world built on D amino acids and L sugars would work just as well in theory. It would be a perfect mirror image of our own biological world. The physics and chemistry would be the same. But that world doesn't exist. We live in an L-amino acid, D-sugar world. This universal preference is called homochirality. So how did this happen? How did life make such a crucial decision so early in its history? And how did that decision become the absolute law for every living thing that followed? There are a few major ideas, and they range from pure chance to the fundamental laws of physics. Let's start with the simplest explanation. It's often called the frozen accident theory. The idea is that the choice was completely random. It was a coin flip. Billions of years ago, in the primordial soup of early Earth, molecules were constantly forming and breaking apart. Self-replicating molecules, the precursors to life, were beginning to emerge. Maybe the very first molecule that managed to make copies of itself just happened to be built from L-amino acids. Or maybe it just happened to use D-sugars in its structure. Once that first replicator got going, it set a powerful precedent. It would need more building blocks of the same handedness to make copies. It would start using up the L-amino acids in its local environment, creating a demand for them. Any other replicator that tried to use D-amino acids would be at a disadvantage. It wouldn't be able to use the same raw materials. Think of it like building with Lego bricks. Imagine you have a bin full of two types of bricks. They look identical, but one has the studs on top slightly to the left, and the other has them slightly to the right. You could build a tower with either type, but you can't mix them. A left-handed brick won't snap properly onto a right-handed one. The structure would be weak and fall apart. Proteins are the same way. They aren't just simple chains of amino acids. They fold into incredibly complex three-dimensional shapes. A common structure is the alpha helix, which is a spiral, like a corkscrew. A protein made of all L-amino acids will naturally form a stable, right-handed spiral. If you try to stick a D-amino acid in the middle of that chain, it would be like a brick that doesn't fit. It would break the spiral. The protein wouldn't be able to fold into its correct shape. And if it can't fold correctly, it can't do its job. An enzyme with the wrong shape can't bind to its target. A structural protein with the wrong shape would be weak. So, the frozen accident theory suggests that once the first successful life form picked a side, L-amino acids for instance, it created a system that only worked with that one type of brick. Any organism that tried to use the other type of brick was at a huge evolutionary disadvantage. It couldn't eat the existing life forms for food. Its own proteins would be unstable if it accidentally incorporated the wrong kind of amino acid. It was locked out. The initial choice, even if it was random, became locked in or frozen for all of evolution. This is a powerful and simple idea. It explains the outcome we see today, but it's also a little unsatisfying. It feels like shrugging and saying it just happened. It leaves open the question of whether there might have been some underlying reason, some subtle push that made the coin land on one side instead of the other. Maybe it wasn't a 50-50 coin flip after all. Maybe the coin was slightly weighted. This leads to the second set of theories, which suggests that the choice was not random. Instead, some external force or asymmetry in the environment of early Earth created a small imbalance of one-handedness over the other. It might not have been a 100-0 split, but maybe just a 51-49 split. But in chemistry, a small advantage can be amplified over time until it leads to a total takeover. One of the most popular candidates for this external force is light, specifically circularly polarized light. Light is an electromagnetic wave. Usually the waves vibrate in all directions perpendicular to the direction the light is traveling. But light can be polarized, meaning the waves are filtered to vibrate in only one plane. This is how polarized sunglasses work. They block the glare that reflects off horizontal surfaces. Circularly polarized light is a step beyond that. The direction of vibration actually rotates as the light wave moves forward, tracing a spiral or corkscrew path. This light itself is chiral. It can be left circularly polarized or right circularly polarized. Where would this chiral light come from? Not from our sun, which produces unpolarized light. But in regions of massive star formation, the light scattering off interstellar dust clouds can become circularly polarized. Supernova remnants like neutron stars are also thought to be sources of this kind of light. The idea is that in the early solar system, the dust and gas cloud that would eventually form the Earth and other planets was bathed in this circularly polarized light. Here's how it could have influenced the molecules of life. Chiral molecules interact with chiral light differently. A beam of left circularly polarized light might be absorbed more strongly by L-amino acids than by D-amino acids. When a molecule absorbs high-energy ultraviolet light, it can be destroyed. So, if an area of space was illuminated with left circularly polarized UV light, it could have selectively destroyed more of the L-amino acids, leaving a slight excess of D-amino acids behind. Or the other way around, depending on the type of light. Over millions of years, this subtle, steady process could have turned a perfect 50-50 racemic mixture into a mixture with a slight surplus of one enantiomer. This is not just a wild guess. Scientists have done experiments in the lab showing that they can use circularly polarized light to destroy one-handedness of a molecule slightly faster than the other, creating a small imbalance. The evidence for this gets even more compelling when we look at meteorites. The Murchison meteorite, which fell in Australia in 1969, is a famous example. It's a carbonaceous chondrite, a type of meteorite that is like a time capsule from the early solar system about 4.5 billion years ago. It has been studied intensely because it contains organic molecules, including amino acids. And scientists found that some of the amino acids in the Murchison meteorite show a slight excess of the L form, the same preference that life on Earth has. This is a huge clue. It suggests that the bias for L amino acids may have existed in space before the Earth was even fully formed. These amino acids could have been delivered to the young Earth by meteorites and comets, essentially seeding the planet with a pre-existing bias. Life, when it emerged, would have started with a pool of building blocks that was already tipped in favor of the L form. From there, chemical processes could have amplified this small advantage until it became the absolute rule. But there is an even more fundamental and more mind-bending idea about where the initial bias came from. It might not be from light or any other environmental factor. It might be woven into the very fabric of physics itself. In the world of particle physics, there is a concept called parity. Parity is the idea that the laws of physics should be the same for an experiment and for its mirror image. If you watch a video of a game of pool, you can't tell if you are watching the real game or a mirror reflection of it. The balls bounce off each other according to the same laws of motion. For a long time, physicists assumed that all the fundamental forces of nature, gravity, electromagnetism, the strong nuclear force, and the weak nuclear force obeyed this mirror image symmetry. Then, in the 1950s, a shocking discovery was made. The weak nuclear force, which is responsible for certain types of radioactive decay, violates parity. It is fundamentally asymmetrical. The universe, at a subatomic level, can tell the difference between left and right. The decay of a cobalt-60 nucleus, for instance, preferentially emits electrons in a specific direction relative to its spin. If you look at the mirror image of the experiment, it looks different. It looks like something that doesn't happen in nature. This is a profound truth about our universe. There is a built-in handedness at the most basic level of reality. So, could this fundamental asymmetry in physics translate up to the world of molecules? The theory is yes, electrons are involved in the weak force. The electrons that orbit the nucleus of an atom in a molecule are the same electrons. So, the weak force creates a tiny, almost immeasurably small energy difference between a left-handed molecule and its right-handed twin. One enantiomer should be ever so slightly more stable than the other. The energy difference is minuscule, perhaps a billionth of the energy of a typical chemical bond. It's an incredibly small effect. For decades, it was considered too small to have any real-world consequence. But life had billions of years to work with. Over huge timescales and countless trillions of chemical reactions, even the tiniest bias can add up. If one form of a molecule is slightly more stable, it might form slightly more often, or break down slightly less often. This could create that initial 50.000000001% to 49.9999999% imbalance. Once that tiny seed of a preference is planted, chemical amplification processes could take over, leading to the total dominance of one form that we see today. This idea, known as the electroweak theory of homochirality, is very elegant. It suggests that life's handedness isn't an accident at all. It's a direct consequence of the fundamental laws of physics. If this is true, it means that life anywhere else in the universe would likely have the same handedness as us. Any alien life form we meet would also be made of L-amino acids and D-sugars, because they would be subject to the same laws of physics. Of course, this is still a theory and is incredibly difficult to prove. Measuring such a tiny energy difference between enantiomers is at the very limit of our current technology. But it remains a fascinating possibility. So, we have these main ideas: a frozen accident, an influence from space via polarized light, or a fundamental bias from particle physics. It could be one of them, or a combination of them. Maybe a small bias from the weak force was amplified by the polarized light in the nebula that formed our solar system, and this biased material was then delivered to Earth, where a chance event in a particular tide pool finally locked it in. We don't have the final answer, but whatever the origin, the consequences of this choice are absolute. A mirror image world is a fascinating thought experiment. Imagine you met a mirror image version of yourself. They would look just like you, but their heart would be on the right side of their chest. They would write with their other hand. And if you tried to shake hands, it would be awkward. If you tried to share a meal, it would be impossible. If you gave them a steak, which is made of L-amino acids, their D-amino acid-based enzymes wouldn't be able to break it down. It would be like trying to unlock a door with a key. That is the mirror image of the correct one. It has the same shape, but it's backward. It won't turn the lock. Similarly, the bread and potatoes you offered, made of D sugars, would be useless to them. They would need L sugars for energy. You could both starve to death at a banquet. This isn't just science fiction. This biological reality of chirality has profound, real-world consequences, especially in medicine. The most tragic and famous example is the drug thalidomide. In the late 1950s and early 1960s, thalidomide was prescribed to pregnant women as a sedative to treat morning sickness. The drug was sold as a racemic mixture, a 50-50 mix of its left-handed and right-handed enantiomers. It turned out that the two hands of the thalidomide molecule had dramatically different effects on the human body. The right-handed molecule was a safe and effective sedative. The left-handed molecule, however, was a teratogen. It interfered with the development of a fetus, causing horrific birth defects, most notably shortened or missing limbs. Thousands of children were born with these conditions before the drug was pulled from the market. To make matters even worse, researchers later discovered that the two enantiomers could convert into each other inside the human body. So even if you took a pill of the pure, good right-handed thalidomide, your body would turn some of it into the bad left-handed version. The thalidomide tragedy was a brutal lesson in the importance of chirality in biology. Our bodies are exquisitely sensitive to the shape of molecules. The wrong shape isn't just ineffective, it can be incredibly dangerous. Today, the pharmaceutical industry is acutely aware of this. Many modern drugs are developed and sold as single enantiomers. The drug ibuprofen, a common painkiller, is sold as a racemic mixture, but only the left-handed form is active. The right-handed form is inactive, but your body slowly converts it into the active left-handed form. The antidepressant citalopram Celexa is a racemic mixture. The single enantiomer version, escitalopram Lexapro, is more potent and may have fewer side effects because you are only taking the molecule that does the intended job. Developing chirally pure drugs is more expensive, but it can make them more effective and safer. So why are there only two handednesses? Because that's the nature of three-dimensional asymmetry. An object can be symmetrical or it can be asymmetrical in a way that gives it a mirror image that is not identical to itself. There's the original, and there's the mirror image. Left and right, there is no third option. The deeper question is why life chose only one from each pair of options and applied that choice universally. The answer lies in the power of standardization. Life is built on replication, on making copies. To make reliable copies, you need a standard set of parts that fit together perfectly every time. You can't build a reliable machine if half your screws are right-handed and half are left-handed. The machine would fall apart. Life chose a standard. All proteins use left-handed amino acid screws. The DNA double helix is a spiral staircase built with right-handed sugar steps. This consistency allows for the stable, complex, and reproducible machinery of life to exist. The origin of that choice is still one of the great unsolved mysteries. It may have been a random fluke that snowballed into a global standard. Or it may have been guided by the light from distant stars or by the ghostly hand of the weak nuclear force. Whatever the cause, the effect is profound. The world is not symmetrical. Life is not symmetrical. This fundamental bias, this choice between left and right made billions of years ago, is in every cell of your body. It dictates the shape of your proteins, the twist of your DNA, the food you can eat, and the medicines that can heal you. It is a silent, invisible rule that governs all of biology, a reminder that sometimes the biggest questions in science come from looking at something as simple as your own two hands.